Hello and welcome back to Honors Biology. Today we're talking about proteins. All right. So proteins, they are in everything we kind of eat. A lot of meat is high protein. So chicken, fish, that's all very high protein food. Eggs are another high protein food as well. Vegetables contain proteins. Again, they're one of our core macromolecules, so they're in everything we eat. The components of proteins are carbon, nitrogen, oxygen, and hydrogen. Those four elements make up proteins. And we see that again in those four elements make up a lot of what we eat. So let's talk about the protein structure. Protein structure, so the monomer to proteins are what we refer to as amino acids. This is an example of amino acid right here. There's a couple of parts I want to highlight to the amino acid. One is the essential carbon. Remember, carbon is the backbone of life. So an amino acid starts with essential carbon. Off of the carbon, we have three what we call subgroups. One is this amine group right here. Nitrogen with two hydrogens is this amine group. Our other subgroup is a carboxyl group. This is what carboxyl is. It's a carbon double bonded to an oxygen and then bonded to an OH. What makes amino acids different, because we have 20 different amino acids, what makes each amino acid different or what determines them is this R subgroup here or R subchain. This is what determines whether you're going to have lysine, glutamic acid, cytosine. That's what determines what amino acid it is. So every amino acid has those parts: amine, carboxyl, R subgroup, and that's all bonded to essential carbon. So if we keep going in structure, right, monomers amino acid, that's our one. As we join them together, they create what's called polypeptides. Right, so here's one amino acid, right, essential carbon, amine group, carboxyl group, R subchain. When they join together through dehydration synthesis, remember, dehydration synthesis is how we get polymers from monomers. So we have two amino acid, two monomers right here, go through dehydration synthesis, and they form a peptide. This bond between the carbon of the carboxyl group and the nitrogen of the amine group creates the peptide bond. This is called a peptide. As we string together peptides, large chains of peptides are referred to as polypeptides. Remember, many peptides, poly means many. As we keep going with our protein structure, as we get more and more peptides, they go through different stages of structure. When we get enough, we enter what's called the secondary protein structure. There's only two different types of secondary protein structure, and you need to be able to realize what each one's called by looking at it. And they're relatively straightforward. One way the secondary structure will look like is a alpha helix, which is just a helix. It kind of looks like DNA, if you've ever seen DNA. This is what the alpha helix looks like. The other way is through what we call a beta pleated sheet, or just beta sheets. And they just look like sheets of paper. That's all they are. They look like this, right? Just flat sheets of paper that just stack on top of each other. And as you see, all of this bond, like this bond right here, this is that peptide bond that we were talking about. The carbon of the carboxyl group gets bonded to the nitrogen of the amine group. So two different structures for proteins, either an alpha helix or a beta pleated sheet. Now, we can get really large proteins. Proteins can be bonded together into a tertiary, so a third structure form, or even a fourth structure form. That just becomes a jumbled kind of mess with a lot of different parts. as you can see, these blue parts here that are rotating, that's all of the alpha helixes. So the alpha helixes form one part of this protein. These purple strands are the beta pleated sheets. So again, right, different proteins come together to form a larger purpose and a larger goal. And all of these strands are just long chains of peptides. So this is what we would refer to as a polypeptide, a protein. Now, one of the main functions of proteins, one of the most important things proteins do for us is they function as enzymes. Enzymes are proteins that help lower the energy needed to run different types of reactions. So if you think back to carbs, we have long chains of carbs, like lactose is one, galactose is one, right? And when we eat those get broken up, they go through hydrolysis to break them apart into their smaller subchains so we can easily digest it. Enzymes help us break apart those larger molecules like lactose. And this is kind of what it does. It helps lower the activation energy. So for us to normally break apart lactose, we need a huge amount of activation energy just to break it apart into the different terms. If we have an enzyme, enzymes lower that activation energy a ton for us to be able to more easily break it apart. Enzymes work through what's called an induction fit model or an induced fit model or a lock and key fit model. And this is what we were talking about by the lock and key. Every molecule has a certain shape to it. Maltose, lactose, they all have a certain shape to it. The enzymes are designed to fit that specific molecule there. So maltose is going to come in, fit perfectly into the enzyme. The enzyme is going to run through hydrolysis, be able to break apart maltose into glucose and glucose. And then the glucose is released, and then the maltase enzyme is free to run off and find again. Some terms you'll hear through this process, right, is the maltose, the sucrose, whatever is sucrose, whatever the molecule is, the bigger disaccharide, we refer to that as the substrate. The substrate comes in and is going to want to fit into the enzyme active site. So the site where the substrate joins the enzyme is called the active site. When the substrate fits into the active site, we call that the enzyme substrate complex. Enzyme, substrate, together, enzyme substrate complex. It runs through hydrolysis here, and then the products that are released are glucose and fructose. The enzyme then floats away to go do something else or do it again. One of the key things to realize here is the names matter. So maltose is the sugar. The enzyme that breaks down maltose is called maltase, ASE ending. Amylose is a common sugar found or common saccharide found in carbs. The protein that breaks down amylose is amylase. So if it's the ASE ending, that refers to the enzyme that breaks down that sugar. Proteins also serve other functions. They're huge building blocks for bigger molecules to join. DNA and RNA are a form, right? DNA and RNA make proteins. All of our enzymes are proteins. Hormones are proteins. Blood clotting that starts with proteins. Proteins are the one that helps initiate that. They transport proteins, so from our gut to get from our stomach to wherever they need to go. They help do that, help fight infections. They serve a million different reasons. So that's all I got for proteins. If you have questions, remember to jot them down, ask me in class, and I look forward to seeing you all next time in class. Have a great day. Bye.